Hello biologists, this is Miss R, and today we're going to talk about nucleic acids and proteins. First of all, I just want to make a couple distinctions. Nucleic acids, answer the question, got DNA? Well, yeah, nucleic acids are DNA. This is a model of DNA, so yes, I have DNA. And you can see that the thing that's different in DNA is the middle, or the bases. There are four colors here in this model, which is of course much bigger than real DNA. Four colors represent the four different bases in DNA, and you can see how DNA is kind of a curlicue here. Proteins, on the other hand, I'll use my necklace here as an example, are made out of amino acids, which kind of you can think of it as like beads on a string. They line up, here's a beaded necklace I cut to be like a string. They line up and they bond to each other and curl up in very specific shapes for their job. So some DNA might look like this, just kind of a or some proteins might look like this. Some proteins are really complicated and have side bonds, and there's a couple different subunits and they look like this. But DNA is always going to look the same. It's always going to be this double helix, this spiral. So now that we've got that out of the way, let's get started with the simulation. Hello biologists, we're going to talk about nucleic acids and proteins. Right here, this is lab 209, nucleic acids and proteins. So you should have your lab sheet out and be writing things down and pausing the video as we go along. We know that DNA and RNA are nucleic acids. That's polymers made of monomers called nucleotides. Nucleotides are the chunks, the sugar, the phosphate, and the base that make up the molecule. They're kind of like putting Legos together or links in a chain. DNA is a long molecule found in all living organisms. You've got DNA, all of your cells do, well, except your red blood cells. And so do all the other organisms that are considered to be living. We can take a look at DNA here and see some of the key concepts. You want to see the base here. This is what's different between all the nucleotides, and this is where the hydrogen bonds form to keep these two strands together. You see how there's a purple strand here and a yellow strand kind of wrapped around each other? They stick together with hydrogen bonds between these bases. And there are four different kinds of bases. We'll scroll down here a little bit so you can see the four DNA nucleotides that make up DNA. Those are those four colors you always see in DNA models. So we want to kind of explore this structure a little bit because we're going to answer this question on your lab sheet. The order of the nucleotide monomers in DNA carries genetic information. So it's the order that you put these A, T, G, Cs in that carries the information in DNA. It's like a binary code. And if we say highlight monomers here, or highlight these four different nucleotides here that have four different bases, you can see this is just half of a DNA strand. You can see what order the monomer is in here. Number 1 is red, so that's C. Number 2 is red, so that's C. Number 3 is blue, so that's A. So the first 3 letters of the sequence are C, C, A. I'll let you figure out the rest. You can pause the video. Nucleotides are the smaller units that go together to make up DNA. Remember we talked about they come in 4 colors. And the reason that there are 4 colors, there are 4 different bases. We can see if we click through here, this part's always going to be the same. Here's the phosphate and the sugar. If we click through here, we can see it's this part that changes. It's the base that changes. And if we do this, it helps us look, and just You notice the green and the purple, keep your eye on the green and the purple, are always going to be the same. But this is 2 rings and 2 functional groups sticking out there. This is 1 ring with 3 functional groups. And here's 2 rings, but just 1 thing sticking out here. So the things that remain the same are these 2 things. And the things that carry the information, the order of the bases, is what carries the information. So pause the video and make sure you have questions number 1 done in lab 209. So now we're on page 2. I just clicked on 2 here. And this is question 2. So the first thing we have to do is figure out how these bases fit together. There's 4 that make up the sequence of the DNA, and we want to know how they stick together. Let's take one of these bases and try to hydrogen bond it to other bases. It doesn't stick so well with G. Oh, look, with T it makes 2 hydrogen bonds. Doesn't really make any with G. So let's put this one with T. Let's take C here. Not going to stick to itself. Makes 1 with A. How about G? Ooh, makes 3 hydrogen bonds there. Let's stick it there. How about G? We've got C and we've got A. Let's put it here. It made 3 hydrogen bonds there. And it only made 1 with A. So T must, of course, go with A. And you can see that there's a pattern here. A and T are always together. So here's A and T, here's A and T, here's G and C, here's G and C. And you can pause the video and count the number of hydrogen bonds. Hydrogen bonds are always represented, well, at least in this particular simulation, as dotted lines, because they're bonds we know that occur between molecules, not within molecules. They're not actually sharing electrons, they're just stuck together by charges like magnets. So you already know this. Predict which pairs you'll find paired up with each of the 4 bases in the DNA double helix. Just so you can see how the helix actually has 2 strands, and those bases come together in the middle and hydrogen bonds form. They're represented, the hydrogen bonds are represented here by the slots here. And it's not really a bond, they're not sharing electrons, they're just stuck together by magnets. A better way for the model would be put together with magnets. So now we know that 2 DNA strands wrap around each other to make a double helix. It's like sides of a ladder. If you took a ladder and twisted it, it would make a double helix. You can see if I untwist this model, it looks like a ladder, and then I can twist it up and it looks more like DNA. So let's translate that to this molecular model here. Here's the space-filling atoms, the atoms taking up space with their electrons. That's not especially helpful, so let's kind of mess around with some of the other models. We can do sticks, we can do balls and sticks, which represents the atoms and their bonds. We can do just a ladder to simplify things a little bit, where the sugars and the phosphates, the things that are the same in all the nucleotides, are just represented as yellow, and then you can see the bases in there. That's helpful for showing the hydrogen bonds. See if I twist it around. You can see all those dotted lines there between the bases. That's a good way to look at the hydrogen bonds. We can go have it in 2 chains. That really highlights the hydrogen bonds there. You can see how the bases are stuck together by those dotted lines, those hydrogen bonds that we looked at, where G is with C and T is with A. And we can look at the nucleotide view as well and see all those hydrogen bonds too. We can see that the blue A is always with yellow T. So there's A and T with 2 hydrogen bonds between them. So that should help you answer the first part of question 3. You can just take a screenshot of my model here showing all these lovely hydrogen bonds for number 3, or you can just describe it if you can't take a screenshot. So in the process of reading DNA, you can make a protein. It's a process where we will study later in the year, where you read DNA, the cell reads DNA and it makes proteins. It makes these complex molecules that do a lot of different jobs in your body and that have very different shapes. Remember, DNA always has that double helix shape, where proteins can have some pretty wild shapes depending on what kinds of things that they do. They can be kind of globby or circular or have holes in the middle and look like pipes. Let's ask ourselves, what are proteins? Here's a big globby protein called myoglobin. And they're going from the ball and stick from the space filling model, kind of a chain model to help you see how all those amino acids are stuck together. Amino acids are the parts. They're like the beads in a protein chain. And sometimes they'll color code them. Myoglobin here happens to carry oxygen in muscle cells. It's one of thousands of proteins we rely on for life. Here's another one that we rely on for life. It's a piece of protein that can grab onto DNA and hold it in place where your mechanisms in your body are reading that DNA to figure out the code in it so they can make proteins. It can turn that protein coding gene on and off. So those tongs are important in grabbing onto the DNA, holding it, and turning it on and off so you don't make too much protein. This protein is called porin. It's found in bacteria, and it makes pores, or makes holes where things can pass through the cell membrane. It's basically a chain kind of woven back and forth to make sort of a pipe. These are all the different amino acids. There are 20 different amino acids that you can put together to make a protein chain. It would be like having this necklace with 20 different color beads, all with different shapes and sizes. So you can see porin has a hole in the middle because it's a pore, it's a hole, it's like a little pipe in the side of a bacterial wall. And all this chain, the green and the yellow and the orange and the red, are made of amino acid sequences. Proteins do have special shapes from their jobs, and the way those proteins get their special shapes is each of the amino acids has a different charge and shape, and as the protein is formed, those charges attract each other. I'm just going to represent the bond by kind of plugging in my side chains with each other, and you can see how this protein starts to take on a shape. This might form a pore in the side of the cell like porin. Other shapes are more globular. Everything kind of sticks together like this shape. All the amino acids form the chain and they all stick to each other, or they make disulfide bonds like this, and they make a very globby shape. So let's look over here. We're supposed to figure out which protein can puncture a cell. Which one kind of looks like it might be designed to stab something? Which one forms a cable? Which one's kind of long and ropey? Kind of like a long, we know how cellulose is a long model and bonds to itself and makes long stringy things in plants. That's a polysaccharide. Well, there's a collagen molecule that's a protein in your nose that does very similar things. And which one's a pore? Which one can act as a channel or a pipe in the side of a cell. You can figure it out. Make sure you mark it on your lab sheet under question 4. So hopefully we're starting to understand that like DNA is made of nucleotides, proteins are made of amino acids, and there are 20 different kinds of amino acids. Here's a better representation because this is more different kinds of beads, or this one has more different kinds of beads as well. So let's play this movie and take a closer look at the links in a protein chain. We're going to look at porin, and we're just going to look at a small piece of the chain, just a few amino acids stuck together. So there's 5 amino acids in just this little part of porin that we're looking at. They stick together, and the part that's the same on every amino acid is blue. They're coloring that blue. The part that branches out here is called the R group, or side chain, and you're going to know that. You need to know that for question 5. The thing that's different in amino acids is the R group, the chain that sticks out. That gives amino acids their different personalities and helps proteins bond into different shapes. They can make bonds, or they just have associations because they're charged. So let's look at a couple different amino acids down here. Let's look at the different options to display them. Here's serine. This is pretty simple. Here's valine. Gets a little bit more complicated. Here's histidine. The different part here is a ring. Let's look at the side chain only. We're going to black out the parts that's the same. And you can see how serine is a little bit simpler. Valine has more atoms here, and histidine gets even a little bit more complicated. Now we're going to look at the charges, because that helps those amino acids stick together or just associate and make bonds, and that helps the protein take its shape. So let's turn surface charges on. And you can see from the surface charge kind of decoder down here that there's some with slight negative charges, there's some with very positive charges on histidine here, and valine doesn't have any charge, it's nonpolar. Let's take the surface charge off so you can see what's under there making the charges. Nitrogen seems to be making those positive charges. Serine, the oxygen, is a slightly negative charge because it's attracting those electrons. And valine is all carbon and hydrogen, so it's nonpolar. It doesn't have any charge. So if we look again, it must be the atoms in the side chain that are making those charges. All right, you have all you need to know for question 5. Here's what you need to know for question 6. We're on page 6 here. And you can read all this. You can pause the video and read all this. Here's our non-polar amino acid valine from the last page. And we're going to look at how valine reacts with water here. You can see that show hydrogen bonds is on. I'll turn it off so you can see the difference. Hydrogen bonds are just these little dotted lines, and they're happening between the water molecules. But are they happening between the amino acids or between the amino acids and the water? We can turn off show charge too, so you can see just the hydrogen bonds a little bit better. So it's important to note how these nonpolar amino acids are going to interact with each other and with water. So when we have a whole protein chain here, we have some amino acids that are nonpolar, they're red here, and some amino acids that are polar, that are blue. You can think of blue as polar because polar will like to interact with water. Let's see what happens when we let it interact with water. And remember, most of the insides of your cells are watery, So this is how a protein is going to behave in your cell. It's going to clump up like this one does if it has this sequence of amino acids, because these non-polar amino acids are going to want to interact with each other and not with the water, and the polar amino acids are going to want to interact with the water. Here we can get a different amino acid and show how different ones are going to have slightly different shapes based on how many polar and non-polar amino acids they have. We'll get a different protein here. But you can see in general, the red stuff all globs up in the middle away from the water next to each other, and the blue stuff is next to the water. We'll let this one kind of sort itself out a little bit. This one's going to be a little bit longer maybe. It might, because it's got more polar stuff in it that's going to kind of stick to the water, it's probably not going to glob up quite as much. So it depends on the sequence, depends on how many nonpolar and how many polar. So now you have what you need to answer this question. If there are many nonpolar amino acids in the protein, where do they end up? And you can also explain how the interactions between water molecules cause the hydrophobic amino acids to fold into the center. Stop the video and make sure you answer on your lab sheet. So now it's time to wrap up nucleic acids and proteins on page 7, which is question 7 on your lab, so make sure you're working on your lab sheet. Let's review quickly what the differences between DNA and proteins are. DNA always has the same shape, has 4 nucleotides, and the nucleotides are different because of their bases. There's 2 strands, and it comes together, and those strands stick together because of hydrogen bonds. So DNA is always this double helix shape. Proteins are not made of nucleotides, they're made of amino acids. There's 20 different amino acids, not just 4 like there are nucleotides in DNA. And proteins can make shapes because these 20 different nucleic acids can stick together because of their different charges and shapes. So now let's look at these questions. What causes the 2 strands of DNA in a double helix to attract and wrap around each other? Well, go look at the screenshot you took for question 3, and you'll see those dotted lines, and that will help you figure it out. Which part of a DNA nucleotide carries the genetic information? Well, it's the part in the middle. It's the different part. And it's the part that things are colored for. The 4 different bases are 4 different colors. This side chain here on our model stands for the sugar and the phosphates. They're what bond together on the sides and create the side of the ladder for DNA. The side chain of an amino acid I like to think of as a little different. And I'm Miss R, and I'm a little different. So that's how you can remember the side chains of amino acids. Think of me, your crazy little weird kind of different biology teacher, Miss R. A protein chain is made out of 20 different amino acids and has a distinct shape. We have this little model. We have ones that kind of glob up. We have ones that have different components and lots of different side chains. Proteins are pretty cool. They're very diverse, depending on what they do. There's a structure-function relationship in proteins. Their structure relates to their function. So that's one thing that DNA and proteins are different. They have different structures because they have different functions. DNA structure, always the same. DNA in any organism is going to have the double helix. Doesn't matter what the code being carried is, the shape is going to be double helix. Proteins, it depends on what their job is. Some of them are going to be like a pipe in the side of the cell membrane. Some of them are going to be kind of squashed up, some up to be an enzyme. It just depends on what they're doing. Some of them are going to be kind of long and ropey, like the protein collagen that you have in your nose and your ears. It's a strong, fibrous protein, also used to make ligaments. So the shape of a protein is going to depend on what its job is, what its function is. And remember, proteins are made out of amino acids, 20 different amino acids. Proteins, amino acids, N, amino acids, proteins. Bases, there are only 4, DNA, or nucleotides. DNA is made out of nucleotides, which is the base, the sugar, and the phosphate. There's only 4 nucleotides. So those are some important differences between DNA and proteins. Last but not least, this protein here is shown folded in water. How do we know which color stands for hydrophobic? That's non-polar. And how do we know which color stands for polar or hydrophilic or water-loving? Water here is in the black. You can consider all of this black area the inside of the cell where there's water. I'll let you think about that one. So we've come to the end of the lab. Thanks so much. I know this is hard work, but you're going to learn a lot, and it's going to pay off in Chapter 3 when we start to use all of this to understand more about the cell. Thanks so much. This is Miss R signing off.